Welcome everyone. I hope you've replenished your stomachs and that you're ready for the next session, which will be Science for Society, as I guess all science should be, and how this Science for Society transforms the present. We have a lineup with a keynote address, and then 4 researchers that are either actual members of CIIMAR right now or have been in the past. There will be a short time for questions after each talk. And without further ado, I would like to invite the first speaker, Professor Alex Rogers, who is a Deputy Director Strategic Science Programs and Partnerships at the National Oceanographic Center in the United Kingdom. He's a marine ecologist with over 30 years experience, interested in how biodiversity is distributed in the ocean, especially in the deep sea and mesotrophic tropical coral reefs. He has vast experience, worked with governments, intergovernmental, non-governmental associations, organizations, on human impacts in the ocean and how to manage these human activities to mitigate or reduce the harm that they might do to marine ecosystems. This has included work on the development of the sustainable blue economy in low and mid-income countries, and advice to negotiators of the BBNJ agreement. Alex is leading strategic development of science at the UK's National Oceanographic Centre. So thank you for accepting our invitation, and welcome, Alex.
Thank you very much, Pedro, and thank you to Joanna and everyone for inviting me to talk to you today. So it's my job to kind of kick off the science here, and I'm going to talk about my favourite place in the world, which is the deep ocean. So the deep ocean was originally a very scary place for humans. In fact, it was considered to contain the debris from the great biblical flood, the one that, of course, Noah escaped from with all the animals. And so it was thought of in medieval times as a kind of evil place. The Old Testament refers to a colossal creature of the deep called Leviathan. And there's a sort of general fear of the deep sea amongst many people around the world during these times. For some indigenous people, the deep sea is still a place where ancestors are thought to dwell. But really, our scientific understanding of the deep sea began in the 17th and 18th and 19th centuries, and one of the first big scientific expeditions to really investigate the deep ocean was the Challenger expedition, which is about 150 years old as we sit here and speak. The really critical thing about the Challenger expedition was that a lot of thought was put into actually working up the results from that expedition. So it circumnavigated the globe and produced over 50 volumes of scientific results from that expedition, including a lot of biology. And this was the main tool of that biology here. This is the famous or infamous naturalist dredge, and this was the equipment that was used to sample much of the life during the Challenger expedition. And something like 4,700 species were actually discovered as a result of the work on this expedition. Now we use much more sophisticated technologies to investigate the ocean. I personally had the luck to work a lot with submersibles and ROVs in the deep sea. These are technologies which actually really were derived from exploration for oil and gas in the North Sea and then in deeper waters. So, in fact, a lot of our modern technology for investigating deep ocean really came as a result of development for industry. And this is our current picture of what the deep sea looks like. And the important thing to note here is that it's not all just flat plains of mud. There are, of course, the mid-ocean ridges, which kind of stitch the earth like a baseball with the seams in a baseball. There are seamounts, mountains, underwater plateau, almost as many geomorphological features as the land. So here off Portugal, for example, we have these amazing canyon features. We've got, as I said, seamounts. This is our own seamount off the UK, the Anton Dawn seamount. The summit of that is somewhere between 400 and 200 metres. And then we have trenches as well in the ocean, the Marianas Trench here, which goes down to nearly 11,000 metres. And for the first time ever, we are now able to regularly put humans down to these fantastic depths using a submersible actually developed with private funding rather than government funding. So things are really, really changing. And also, right on the, it'll be on your right-hand side here, you'll see some maps of parts of the East Scotia Ridge in the Antarctic. And I'll actually come back to these in just a minute. But these are parts of that ridge which are kind of swollen up. So we could map those areas and then home in on them with our biology. So these new technologies have really revealed some fantastic new animal communities in the deep ocean. This is an example of one that we discovered in the southwest Indian Ocean on a seamount at about 1,000 metres deep. Now, interestingly, the fishermen in the area had named this seamount Coral Seamount, and here you can see why. This is an amazing cold water coral reef sitting at 1,000 metres. Now, I can tell you, as we were going across this with the ROV, we could see in the lights of the ROV hundreds of pairs of eyes peering out from all the cracks and crevices. These are all the crustaceans living in amongst this framework. And we found many, many new species on these seamounts during the expedition. This is the first high-temperature hydrothermal vents found in the Antarctic, and these were located in those swellings on those ridges that I spoke about. These were not the animals we were expecting to see when we went here. We were expecting to see a mixture of fauna from the Pacific and the Atlantic and so on, but instead what we found was a completely new fauna. And as you can see, there were rather a lot of these crabs. These are the famous Yeti crabs. They have very hairy chests. So one of my PhD students nicknamed them the Hoff crab after David Hasselhoff from Baywatch. But they actually grow bacteria on these hairs and make their living by scraping the bacteria off and consuming them. But the interesting thing here was that for a single expedition of 4 weeks, we completely changed the idea of the biogeography, the distribution of animals on the hydrothermal vents globally. So this is a really amazing thing that a single cruise or expedition can really change our ideas about the deep sea. They do spend a lot of time grappling with each other, these crabs. It's quite interesting. So they're big animals, of course, but most of the diversity in the deep sea exists in much smaller class of animals. The macrofauna, the sort of worms about the size of an earthworm that you can find in the ocean, and the myofauna, the really small animals which live amongst sand grains and grains of sediment in the deep ocean. And it's in these sediments and on them that these really fantastically high diversities of animals occur. And this really lovely photograph here from Gilles Martin in Ifremer just gives you an idea of some of the diversity that is found in these deep ocean sediments. Now, of course, as scientists, we're still only just exploring the deep ocean. Unfortunately, we've been exploiting it for a considerably longer time. And this is just one example. Deep sea trawl fisheries, which have been operating in the deep sea since the 1960s. And as we can see here, this is a fish species called the orange ruffy, one of my favourites. And when these were first located, spawning over seamounts, they were looked on as an incredible resource for fisheries. And you can see some examples here of these apparently very clean catches being brought on board. These are off New Zealand and Australia. But what wasn't known when these fisheries commenced was just how long-lived these animals are. These fish can live to be over 150 years old. They don't mature until they're 30 or 40 years old. They aggregate around seamounts to breed, so they're very vulnerable to high levels of fishing, and they've been targeted globally in temperate waters. And of course, these features make these fish highly vulnerable to overexploitation. And what we saw in many cases were stocks of these fish being collapsed very rapidly after commencing fishing, including off Europe. What became apparent in the 1990s were that these fisheries were associated with other impacts. And this was the pair of photographs which really switched me on to this topic. So what you see here are photographs from seamounts south of Tasmania, and the seamount on the right-hand side here has been trawled for orange ruffy and oreo, and the one on the left was too deep for the trawling. And you can see the really stark difference here. Essentially, when these fisheries commenced, they were associated with a high bycatch of cold water corals and other organisms. Of course, that bycatch tailed off as essentially these ecosystems were completely obliterated by bottom trawling. And what we've also discovered since is that some of these animals are incredibly long-lived. So some octocorals and black corals have been discovered to live for thousands of years, up to 4,000 years. And there's even evidence that a deep sea sponge, which occurs in the South China Sea, can live for up to 12,000 years. And I believe there's some new work on the way to show that coral longevity actually extends even beyond the 4,000 years we have at the moment. So the rates of growth for these animals is incredibly slow. Their recruitment relatively low, and we've seen very little evidence of recovery of these sorts of impacts on these deep sea ecosystems. So the scientific evidence really was taken to the UN General Assembly, and it resulted in a number of resolutions to try and increase sustainability and improve the management of these deep water fisheries. And in 2008, these resulted in what were called the International Guidelines for Management of Deep Sea Fisheries in the High Seas. I was actually on the IUCN team at these negotiations, and again, I can tell you that the scientific evidence presented at these negotiations was absolutely critical to get these guidelines moved forward. So now we're moving really into a new era of conservation of deep sea ecosystems in the high seas or areas beyond national jurisdiction. And that is because of this new biodiversity beyond national jurisdiction agreement. So you can see in dark blue here are all these areas beyond national jurisdiction. It's about 45% of the Earth's surface, 54% of the sea floor, but 95% of the living space of the ocean. Now, the BBNJ agreement has been developed over more than 20 years of negotiation. And the actual conferences to try and negotiate the text for this agreement were really quite hard fought. But now we think that by the end of this month, September, there will be over 60 ratifications for this agreement, so it is likely to come into force before the end of this year. And I think it's about 18 months after those ratifications take place that it comes into true legal force. Now, this agreement is focused on the sharing of marine genetic resources, but for the first time, it sets up a legal framework to protect marine biodiversity in this vast area. So it also looks at area-based management tools, things like marine protected areas. It sets up a regime for environmental impact assessment, and it talks a lot about capacity development. So one area that we've been looking at with potential impacts in the high seas is, of course, deep sea mining. And I wanted to focus a bit on this talk because it really shows the importance of science when thinking about human activities in the deep sea. Deep sea mining is not just about polymetallic nodules, those potato-like things you see on the right-hand side. There are many other types of mineral deposit in the ocean. There are phosphate deposits, potentially used for fertilizer, seabed massive sulfides associated with hydrothermal vents, cobalt crusts on seamounts, and muds which are rich in rare earth elements. And these are now becoming strategically important metals to high-tech industries. Now, of course, there are potential impacts from deep-sea mining should it take place at an industrial scale. You can see the size of the kind of crawlers which may potentially be used to exploit these minerals, huge machines which hoover up or suck up manganese nodules, for example, and there are a whole range of different impacts which may be associated with these activities, from impacts on the sea floor to the release of sediments into the water column to noise and other aspects. So these mineral deposits are located at various places around the world in the deep sea. You can see this area on the left-hand side, these kind of purple blobs. That is a huge area of these polymetallic nodules. Then there are seabed massive sulfides, you can see with the red blots here on the mid-Atlantic ridge, also in the Indian Ocean, and finally the cobalt crusts in orange, and there's a particular area for those in the north-west Pacific. So at the National Oceanography Centre, we've been working on a project to look at the diversity of the Clarion-Clipperton fracture zone, this vast area in the equatorial eastern Pacific that might be subject to mining. And, the thing to really think about here is just the sheer size of this ecosystem. The deep sea is pretty much everything below 200 meters. That's the volume of about 1 billion cubic kilometers of ocean, an area of 360 million square kilometers, and as I said, something near to 50% of the Earth's surface. So this presents some real challenges, just the sheer scale of this ecosystem. So this is the kind of prospecting area for polymetallic nodules. You'll see it located there in the equatorial eastern Pacific. And these numbered areas here kind of conservation zones to basically preserve areas from mining, but also to look at the kind of baseline of the science, particularly biodiversity in these regions. So this is what all the excitement is about. These are polymetallic nodules. They contain a range of metals. They take millions of years to grow. So this is a non-renewable resource. Once you take them off the seabed, they're not going to appear again in humankind's lifetime. But the important thing to point out here is that those nodules themselves are hard substrate for animals to attach to. And of course, here we see a glass sponge growing on one of these nodules. And the first thing to notice here is the association of other animals living on this sponge. So this is a real case of biodiversity begetting more biodiversity. The animals are providing part of the structure for other animals to live on. And one of the kind of propagating impacts potentially of mining is removal of these biological habitats for other organisms. So, as I said, many species are reliant on hard substrata. And of course, one of the things this work has demonstrated is that the greater the number of nodules, the higher the density of nodules on the seafloor, then the higher the density of animals and the higher the diversity. Nodule type and patch size is also important in determination of the makeup of these animal communities. So not only are the nodules distributed at different densities, but they can have different morphologies as well. And what we find is that patches of a similar nodule type have similar biological communities. Higher density nodules have higher densities of animals. Lower densities have lower densities of animals, but potentially a higher diversity. And then, of course, areas of mixed nodule shapes have the greatest diversity of all. One of the other things this work has shown is that, as I said, people have typically thought about the abyssal plain as these huge, vast plains of mud. But both this work and work in the Atlantic and the Sargasso Sea have actually shown that hard substrata is much more common than we previously thought on these abyssal plains. So there are outcroppings of basalt on these largely what are thought of as flat and featureless areas. Seabed topography is also important. It was found that the Clarion-Clipperton fracture zone is not, as I said again, a flat area. It has small hills, small seamounts, ridges on it, and so on. And these all have an influence on the biodiversity, on the biological communities that live on the seafloor. So this adds to the heterogeneity in the biodiversity of life. There are also large-scale patterns of life in the Clarion-Clipperton fracture zone as well. These are linked to primary production at the surface. So there is a gradient of primary productivity running from north to south. That influences the density and biodiversity of the fauna. But more recently, we've also found that the carbonate compensation depth also has quite a profound effect on the diversity of the communities found. And so where this transition zone is, between 4,300 and 4,800 meters depth, there is a transition in the biological communities on the seafloor. So the ocean chemistry here is playing an important role in driving the biology. So here's a good question. How many species actually live on the seafloor of the Clarion-Clipperton fracture zone? Well, interestingly, about 436 species have been named from this area, but we've recorded 5,578 from the region. So less than 10% have actually been described. Estimates from sampling to sample which have been taken from this region suggest that there's somewhere between 6,000 to 8,000 species in this region. And this really echoes what we know about the ocean in general. Somewhere between 10 to 20% of organisms have actually been described, with all the rest at the moment unknown to science. This is a huge challenge when trying to think about gaining biodiversity baselines for the ocean to really understand what the impacts of human activities may be. This is from a paper from Philippe Boucher from the Paris Museum, and he has estimated it takes on average about 13 and a half years to describe a species once it's been sampled on a scientific expedition. For some groups, like sponges, that number goes up to 24 years. So this is a huge, huge challenge for us scientists. We've really got to bring all our tools of integrated taxonomy to bear on this problem to try and accelerate how quickly we actually describe this biodiversity. And new tools like eDNA are really helping us to start to understand the patterns of biodiversity in the deep ocean. So I thought I'd just finish by saying something about really conservation of biodiversity in the ocean. And the first point I wanted to make was it's more effective to protect than to restore biological communities. It's not only more effective, it's a lot cheaper to actually protect these ecosystems than to try and bring them back after they've been damaged. So the important point to make here, though, is that marine spatial conservation measures, things like marine protected areas, must actually protect species against the activities which damage these biological communities. So it's no good having a marine protected area to protect organisms which are damaged by bottom trawling if the protection doesn't include keeping out bottom trawling from the area. And just a reminder here that the Kunming-Montreal objectives from the CBD now mean that we have to protect at least 30% of representative habitats in the ocean, but also we have to think about restoring at least 30% of those which have already been impacted. So baselines of species and habitat distribution are absolutely critical for managing human activities in the ocean, but also thinking about actually conserving and protecting them. And in areas beyond national jurisdiction, the new BBNJ agreement is the legal framework by which we'll be doing this. The other point I wanted to make, and I've noticed that some of the work here on CIIMAR is really focusing on restoration, is that restoration science for most marine ecosystems is barely in its infancy. We have to do much, much more work thinking about how we can actually restore marine ecosystems, including those in the deep sea, because, as I said, the evidence that they're going to recover without help is actually very limited. So here we can see a couple of examples of attempts at deep sea restoration. One is on Oculina banks off Florida. These are deep water, cold water coral banks. And here they actually place these structures on the seafloor with corals transplanted onto them. Unfortunately, they didn't work very well because people are still actually trawling in this region and destroying these structures. A really interesting example has come from the Mediterranean, which you can see here on the right. This is work done from Spain. And here the scientists are actually working with the fishermen. They're collecting the coral bycatch from the trawlers or the fishing gear, and then they're attaching the corals to natural an artificial substrata, and those can be chucked over the side of a fishing vessel when it's out in the ocean. And this is actually looking like quite a promising approach to restoring these particular corals from this particular ecosystem. So it's a very nice example of where working with the industry may help to actually restore the habitats here from damage. But as I said, an important point to make here is that often restoration is much more expensive than preservation. So to summarize that, ocean science has really demonstrated the deep sea is this fantastically rich area in terms of topography, but also habitats, and it's very species rich. Despite our advances in understanding deep sea biodiversity, knowledge is still very limited. Human activities such as deep sea bottom trawling has already severely impacted rich but fragile deep sea ecosystems. The policy response has generally been reactive, but has advanced regulation and management of activities in the deep sea. Implementing policy such as the BBNJ agreement is really going to require a lot of input from science. So we're going to need a lot of baseline data about biodiversity in the deep ocean, the distribution and diversity of both species and habitats. And if anyone's interested, there is a book coming out on the BBNJ agreement in November, and there's a chapter in there which I've written on the science needs of the BBNJ agreement. And then, of course, implementing a global network of spatial conservation measures is really important, but we've already got to think about how to restore the deep sea ecosystems that have already been severely impacted by human activities. And, of course, we have the spectre of climate change also to contend with when thinking about both restoration and conservation. So with that, thank you all for listening. A special thank you to Dan Jones from NOC, who allowed me to plunder his picture library for this talk. And yeah, open to questions, I guess.
Okay, so we have time for a couple questions from the audience, hopefully. Katarina?
Thank you so much, Alex, for your presentation. It was really inspired me. I have a curiosity about these sessile species that grow in these mineral nodules. So they are endemic species to these regions, or they also are able to grow in another kind of substrate or areas in the deep sea?
That's a really tough question to answer at the moment. I have no doubt a lot of them probably attached to any hard substratum in that CCFZ area. We have seen, for example, in the seamounts in the Northwest Pacific, there are differences in the communities on hard substrate covered in cobalt crust versus substrate which is just bare basalt. So there does seem to be some evidence that probably the chemistry of some of these mineral concretions is having an influence on what attaches to them. Certainly in the case of cobalt crust, and I guess it may be the same with these manganese nodules. And of course, the other thing that has just been discovered and is being looked at the moment is the possibility that these nodules are actually generating oxygen. And they may also be generating hydrogen as a result, and essentially this oxygen generation may be occurring, and I say maybe because we're not sure yet, but it may be occurring because of the splitting of water in a form of electrolysis. if that's going on, I think that's likely to have an influence on the communities as well. In terms of whether the fauna is endemic to this region, that is a really difficult question because we've got very little understanding of the range of species in the deep sea at the moment. There have been some really nice eDNA studies recently, which suggests that certainly different oceans have different biota, but how that translates down to the regional level is not clear at the moment. One thing that study or those studies have thrown up is, of course, this vast number of sequences to which we can't actually attribute actual species or organisms. And that's not just at the species level, that goes right up to the level of orders and maybe even higher. So this is just more evidence that in terms of our understanding of the biodiversity out there, we've only really scratched the surface. Thank you.
There's a question up front here, Bill Gerwick.
Mining of deep sea methane hydrates and where we should go or maybe not go in that area.
My instant reaction to that is we really don't want to be digging more hydrocarbons out of the ocean. As a scientist, I think it's our job to essentially provide the evidence that policymakers need to really look at the options in those sorts of contexts. At the moment, I would say we probably don't have a brilliant understanding of how diversity is associated with these methane hydrates. We have a paper just about to come out on the discovery of the world's deepest methane hydrate site in the Arctic, just north of Molloy Deep. And the interesting thing about that system is that it seems to share communities with the Arctic hydrothermal vents rather than with the shallower methane seep sites. So there's something interesting going on there in terms of biogeography. So I'd say there is, on the biodiversity side, still a lack of information on biodiversity. But as I said, I think any form of new hydrocarbon exploitation has to be looked at in the cold light of climate change.
Agreed.
Yeah.
Okay. There's time for one more question.
Thank you for this presentation, and not being myself expert on deep sea, I'd like to ask you about, and I heard some presentations about this production of oxygen in the deep sea. If this is really something that we need to consider also when trying to discuss about this exploitation of minerals from the deep sea.
Absolutely. I mean, we already know that the polymetallic nodules provide habitat for organisms, but if it turns out to be correct that these manganese nodules are almost acting like mini batteries on the seafloor and are electrolyzing seawater to produce oxygen and hydrogen, that really throws this ecosystem into a whole new light. I've done work in the past on thinking about the impacts of changes in oxygen concentration in the deep ocean. And we know that in deep time, there are periods when oxygen has been severely depleted in the deep sea, possibly even complete anoxia for some basins. But if it was the case that these nodules are there producing oxygen, who knows, that may have been a refugia during these times. So it has implications in terms of our understanding of the history of biodiversity in the deep ocean. If they are generating oxygen, then and hydrogen, because hydrogen, of course, is a chemical substrate for chemosynthesis, then this must be significant in terms of the biology. And of course, if you take away the nodules and you reduce that function, then you're not just talking about taking away the hard substratum, but also altering the chemistry of these deep ocean environments. Now, as I said, this was a preliminary finding. There is work ongoing at the moment in the Scottish Association for Marine Sciences on trying to confirm whether or not this really is the case. So, yeah, we're all excitedly waiting to hear the results of those investigations.
The last question goes to our Chief Commissioner, because she kind of forced me to let her ask a question.
I'll try to be quick. Thank you so much, Alex. Lovely to hear you again. You alluded really well to the sheer vastness of the deep ocean. And I think we're at a point in history, a very exciting point in history, that we have full depth observation capacity at a global scale. However, this capacity is very inequitably distributed. And a recent study has also shown that we've only actually observed 0.001% actually of our deep sea floor. So how do you see us going into the future? How can we actually expand and accelerate these observations, especially for countries which have more limited resources and or infrastructure for such observations?
Yeah, that's a really good question. There are probably many parts to answering that question, but I'll probably home in on two things. First of all, technology is going to provide us with a lot of help in this area. We're seeing the distribution or the development now of really quite large and sophisticated fleets of autonomous platforms. Those autonomous platforms are now carrying equipment that can, for example, sample environmental DNA over the period of time that the platform is in the water. So we can potentially get a picture of the kind of genetic diversity of life in a much better way than we have been able to previously. There are also developments in things like hovering, AUVs, a whole variety of different flavors of these autonomous platforms that are going to help us. What I would say, however, is that not in the next, I would say, 4 or 5 decades, will that technology get to the point where it can efficiently sample actual organisms from the seafloor in a way that allows taxonomists to actually work on that material. So for the foreseeable future, I see a combination of autonomy to cover large areas, but still the need for ship-based sampling for this kind of baseline work. And also all the other ecology work that you have to do to understand food webs, to understand how these things reproduce in deep ocean, and so on. On the capacity side, and I talk about this in this book chapter on the BBNJ agreement, we've got to move to a way of co-production of science with low and middle income countries. It's no longer good enough just to say, oh, we've got 5 places on our cruise, let's send some people from wherever it is to come participate in the expedition. We have to really make this science open to these countries. They've got to be involved in planning the science, actually going out and doing the field work, actually working up the material in the lab and writing the papers, and then translating that knowledge to policy in their own countries. The fascinating thing about the BBNJ agreement is that there is such an enormous requirement for scientific knowledge to drive the agreement, that we have not got the capacity to deliver that in developed countries. So here, there is actually an absolute need to build capacity in low and middle income countries to do this kind of work, to ensure that we've got the human power to actually deliver the science needed for the agreement. So,
Thank you very much.
Thank you, Professor Rogers, for taking us on this trip to the deep sea. Thank you.
thank you.
It's my pleasure now to invite my colleague, Benjamin Costas. He's a principal investigator, group leader here at CIIMAR, PhD in animal science by the University of Porto, and his group, called Aquatic Animal Health, or A2S, has a strongly motivated young team that is working on essentially nutritional immunology. Benjamin is passionate about aquatic animal health and welfare, especially host pathogen interactions and neuroendocrine immune crosstalk. In recent years, Benjamin has been leading two large Horizon Europe grants, Ignition and Green Aqua. And his most recent research is targeting novel strategies to improve fish disease resistance and welfare, as well as non-invasive biomarkers of animal health and welfare. So, Benjamin, the floor is yours. Thank you.
Thank you, Pedro, for the introduction. And I would like to thank the scientific committee for the invitation for the talk here. And I will talk about some tools that we are developing for preventing and mitigating the climate change effects in aquaculture. So I will give you a very brief overview of the thing, and then I will talk about some of the things we are doing in our project Ignition. So basically, we all know our planet is melting. There are huge consequences about climate change effects. Some of them are related to sea level rise, rising temperatures, ocean acidification, changes in rainfall and precipitation patterns, emerging diseases, and harmful algal blooms, and others such as, for instance, jellyfish. All of these have direct consequences in the aquaculture sector because fish are housed and under farmer conditions, and this will force them to lead or to cope with this kind of events, which are extremely stressful for them. As you all know, world population is increasing significantly, and by 2050, world population is about to reach the 10 billion people, which means that there is a significant requirement of animal protein availability. And I don't know if you are aware of this table, but basically, maybe in the back you don't see the numbers, but basically these numbers say that aquaculture is the most sustainable animal production, considering feed conversion rate, water consumption, and carbon footprint, for instance. But the sustainability of aquaculture is at stake at this moment because of the consequences of climate change. I will not go into the details of this infographic, but just to say that there are huge consequences about how contaminants, for instance, or this part here, which is directly related to climate change, such as increasing water temperature, dropping oxygen saturation, or dropping salinity, or the increasing pathogens. This is directly related to climate change effects, and this will force the fish to cope with them, and there are a number of metabolic and physiological responses that interestingly have genetic influence, which makes the animals to be more proactive or more reactive to these consequences. And this opens the opportunity for genetic selection and select animals with most positive traits and make them more resistant, for instance, to pathogens, or make them to grow even faster. And nowadays there is a huge issue in the aquaculture sector, which is the animal welfare. It matters a lot because there is more and more data that are supporting that actually fish have sentience, and there is a demanding need for more research regarding how fish cope with different farming conditions, and more importantly, we need to find tools to assess fish welfare non-invasively or minimally invasively, without the need of touching the animals and disrupt them. So there were some concerns about these kind of issues at the European Commission level, and there was a call in the Cluster 6 within Horizon Europe, where they were asking to reduce the environmental impact of aquaculture by means of minimizing nutrient waste, reducing the antibiotic use or other chemicals used as therapeutic agents, and also to reduce pollution. And that's why we came up with this project, Ignition. This is a project that CIIMAR is coordinating, and we are mainly aiming to use advanced technologies to monitor health and welfare parameters, and also to develop immunity-enhancing feeds, select animals with more resistant traits, not only against pathogens, but also against, for instance, heat waves, and also to prevent diseases using more effective vaccination strategies. And now I will talk very briefly about some of these topics using some infographics that one of our partners did, so you can easily understand what it means, instead of showing you lots of experimental data. And basically, what we are developing functional feeds by means of extracting bioactive compounds from halophytes. Why halophytes? Halophytes, for those not familiar with that, are salt-tolerant plants that can grow in salt environment, meaning that we don't need fresh water to irrigate them, which is a positive thing already, because as you all know, fresh water is precious, and we are lacking fresh water more and more. And this plant, as you can see, has this aspect, but only the green tips are used for human consumption. So it's the edible part of the plant, and the rest is waste. And we can collect this plant as a green biomass or a lignified biomass, and we can use any of this biomass, which is waste, because the green tips were already collected. We are contributing to circular economy concepts, and we can prepare, for instance, a green juice or a fibrous biomass that we can therefore ferment or extract through salt removal cascade, and we can obtain probiotic proteins, bioactive polyphenols, and prebiotic fiber extracts. We are using all these compounds to develop functional feeds, which will allow the animals to have a more robust state, and that eventually would allow them to resist more against opportunistic pathogens, or eventually against heat waves or other effects of climate change. We are already doing that. We already have some prototypes, and we are observing that after feeding fish with this kind of diets, they are resisting more against bacterial pathogens, which is quite interesting and positive. We are also developing oral vaccines with some of our partners, that it consists of genetically engineered bacterial outer membrane vesicles, and basically very rapid, very fast, we are collecting these outer membrane vesicles, we are removing the virulence factors, and then we are using this as platforms of antigen delivery into the host. So basically we are at this step now, we're introducing different heterologous antigens from bacteria and for virus, and we can put a single antigen or different antigens, so we can produce polyvalent vaccines, and the ultimate goal is to use this technology to have adjuvant-free oral vaccines. This has two positive effects. On the one hand, it simplifies the cost of the producers because you are delivering the vaccine through the feed. And on the other hand, it has no adjuvants. And the adjuvants, as you all know, it causes chronic inflammation and it will harm it will affect the welfare of animals. Another technology we're developing under ignition are innovative non-invasive biomarkers to monitor animal welfare. And basically what we are doing is we are performing huge databases using classical biomarkers such as cortisol. All of you know what cortisol is. Discovering new biomarkers from skin, mucus, and water through omics technologies, and operational welfare indicators. What are operational welfare indicators? So those are practical, measurable criteria that we can use, for instance, to assess welfare in animals. This is something that is usually implemented at the farm level, so it needs to be cost-effective and easy to understand and to implement. And for instance, we can measure, as it is exemplified here, changes in breeding patterns. For instance, we can assess behavior and other kind of traits. And then we integrate all this data in a data integration platform where, through machine learning, we are developing some algorithms to be able to predict how animals will face different conditions related to climate change. And this is also allowing us to develop electrochemical biosensors. I'm adding here an example, for instance, of the detection of cortisol. This is something that is already available. It was already being used. But we are going a bit further, and we are using skin patches with microneedles where we can assess in real time the condition of the animals in terms of health and welfare. And we are also developing a floating egg that can assess in the water different parameters related to health and welfare, and we can monitor in real-time conditions how the fish are dealing with different challenges. This allows continuous monitoring, and we can assess multiple parameters with multiplex biosensors that are being developed now. And this is just to exemplify. You don't need to look at this because it's a bit complex, but just to simplify that the project is actually running in parallel with different species. You can see we have from fish to halophytes to invertebrates such as shrimp and clams, and we are developing a number of tools that at the end will allow a number of technologies that can be used by the industry. So by the end of the project, we are contributing to the One Health concept, and we will have we are contributing to healthy environment by exploitation of halophytes, the non-food fraction. So we're contributing to circular economy, use of land not suitable for agriculture, try to grow other crops, and a drop in excessive or inappropriate use of antimicrobials. So we are helping to grow healthier animals, and of course, if healthier animals are available, healthy people will be affected by this in a positive way. So this is our consortium, 17 partners from 8 different countries. Thank you, and open for questions.
Thank you, Benjamin. We have time for one question only. Very well.
Thank you so much, Benjamin, for sharing with us these developments on aquaculture, these new technologies. I would like to ask you if you think that right now, with all these developments and application of these new technologies for aquatic animal production, we will be in the near future in the position where we will be able to limit our impact on collecting wild animals from our oceans? Or do you think, like it happens, for example, for terrestrial animals? Or do you think we are still far from achieving this objective?
Well, thank you for the question, Katarina. Well, actually, animal protein coming from the ocean in aquaculture is contributing for more than 50% compared to fisheries. So usually fisheries was always above aquaculture in terms of captures, but now it's below. And I think this is going to grow even more because we need to think of sustainable ways of producing animals or capturing them. At this moment, fisheries, it's a good option, but it still contributes a lot to greenhouse gas emissions. And in the case of aquaculture, it pollutes less, and therefore it's a bit more sustainable. So I think we will rely more and more on aquaculture products not in the very far future, but in the near future. I hope I answered the question.
Please join me in thanking Benjamin once again.
Thank you.
Now we have a different kind of, the session is called Science for Society, Transforming the Present, and now we're going to talk about a very particular present, which is ancient, and it's Paula Campos, my colleague here from CIIMAR, the principal investigator, has a PhD in ancient DNA and evolution at the University of Copenhagen, where she did also postdoc research and worked as a curator at the Science Museum of the University of Coimbra, then assistant professor, again at Copenhagen Natural History Museum, and later also postdoc there. Since 2009, she's joined us, so another example of someone we retrieved back from the cold countries, and she's been with us for some years now, and her research focuses on marine resource exploitation through time. So present is also involved, particularly how human activities have influenced the population dynamics of explorer marine species. She integrates archaeological, genomic, ecological data, and her group, Palomar, aims to provide long-term perspective on the interactions between human societies and marine ecosystems. So again, thank you, Paula, and we look forward for your talk.
Thank you, Pedro, for the detailed introduction. I would first like to congratulate CIIMAR on its 25th anniversary and thank the committee for inviting me to present my work. So I'm going to talk about ancient DNA. So the first thing we need to know is what is actually ancient DNA. Ancient DNA is basically DNA that is recovered from historical, subfossil or fossil specimens like the ones that you see here in these photos. Most of the ancient DNA studies focus on bones and teeth, but also coprolites, which are just fossilized feces, hair, ice cores and sediment cores that are used for environmental reconstructions. But there are a lot of other organic material that you can get ancient DNA from, like feathers, ceramics, shells, eggshells, and even dental calculus that you can use to look at past human diets and also past oral microbiomes. But ancient DNA field is a quite new field. It started in 1984 when Iguchi and colleagues were able to extract and sequence DNA from an old skin of a quagga, and with that they were able to phylogenetically place this animal as a close relative to the zebra. And in 1994, Haas was able to extract and sequence DNA from a mammoth bone that was retrieved from permafrost. And with these, they unlocked the potential of permafrost as an excellent environment for the preservation of ancient DNA. The preservation of ancient DNA is quite challenging because, you know, like, as the minute after, the second after a plant or animal dies, DNA starts to degrade, first by endonucleases that are inside the cells, and then by hydrolytic depurination, deamination, oxidative damage, fungal and bacterial degradation, and the DNA molecules break into shorter and shorter fragments that are at some point no longer usable for us. So when we do an ancient DNA extraction, what we retrieve, it's not only the endogenous DNA, which is what we are actually interested in, but we also retrieve exogenous DNA from like microbia and fungi that are present in the soil where the sample was collected, and contamination that could be like human DNA from some other researchers or archaeologists handling the samples. So if ancient DNA is so challenging to work with, why do we still use it? Because ancient DNA can tell us a lot about the past that we cannot infer from modern data alone. So it allows us to uncover past ecosystems. It allows us to taxonomically and phylogenomically place extinct taxa based on molecular data and not only morphological data. It also allows us to uncover past species distribution ranges, which are completely different from present ranges. And also track migrations and colonization events, for instance, like the human colonization of the New World. And also to study resource exploitation in the past and through time. We know that the Romans were one of the first societies to fully exploit marine resources. They were able to get from the sea a vital source of protein, fish, fuel, clothes, and other raw materials, and with this they shaped the economic trajectories of European societies. They constructed these fish salting plants all along the Atlantic facade and the Mediterranean Sea of the empire, where they preserved fish for trading, for long-distance trading. Small fish like sprats, anchovies, mackerels, and sardines were considered of low value, so they were used to make sauces. Big fish like tuna were considered super valuable, so they were dried and salted for long trade. There were different kinds of sauces that were produced at the time. Garum and liquamen are the most famous ones, and they're used interchangeably. Basically, it consists on the use of small pelagic fish macerated in the brine and left to ferment. These sauces were then used as a condiment in several different recipes. There are written records of these recipes because they had a really umami flavor. Alec and muria are leftovers of these garum and liquamen sauce production. What can we learn from getting ancient DNA from archaeological fish? Well, for once, we can actually identify which fish species we are dealing with. Because in this case, when we are looking at specimens from fish salting vats, they are mostly broken, so they are very difficult to identify morphologically. And it can also give us open as a window to dietary habits and cultural insights of past societies and their subsistence economies. And in a more biological way, we can learn and we can trace how the fish population dynamics were from the past into present. But there are challenges in retrieving usable DNA from this material. Again, because the bones are fragmented, difficult to identify morphologically, and especially because they were kept in an acidic environment, like a fermentation environment, where ancient DNA degradation is exponential, so it is faster than in other environments. But still, we decided to sample some Roman specimens from a site in Adrovelho in Galicia, a Roman site where there were fish salting vats. And in one of the vats, in vat number 1, in the bottom, we were able to recover ichthyofauna. This ichthyofauna was then sorted by our zooarchaeologist colleagues into spines, scales, opercula, etc. And I was given some putative sardine specimens, mostly broken vertebrae that you see on those pictures. And my aim was to first try to see if I was able to get DNA from these samples and then look at the genetic diversity of this old population, so a population pre-exploitation and overfishing, and ideally to look at effective baseline levels on fish populations. So all the samples that we had from Adrovelho were actually European sardine. we screened 17 samples, and of these we decided to sequence full genomes of 12 of them. And why? Because we had a lot of data from modern sardine populations. We actually have 188 genomes available. This work was done here at CIIMAR and led by my colleague Felipe Castro. So we have 108 genomes from 12 locations across the species distribution range, the current species distribution range. And we know that nowadays sardine divides into 3 different groups: a western group that clusters Azores and Madeira, a central group that clusters Morocco, the Iberian Peninsula, and the Atlantic facade in France, and then an eastern group that joins together samples from the Canary Islands and the Mediterranean Sea, as you can see both in the admixture graph and the PCA plot. So when we analyze our samples from Adro Velho together with these 180 genomes from modern populations, we can see that they clearly cluster with the central group, both in the admixture graphs and in the PCA plot. Adro Velho is in red and here in blue. One thing that is striking is that these populations, the Roman sardines, seem to be less admixed than current modern populations, and this could point to a possible result of overfishing and species exploitation. Another example of how one can use ancient DNA to look at resource exploitation in the past is the case of the Basque whaling in the Cantabrian Sea. There are written records that point that the North Atlantic right whale was the main catch of these whalers, but there was no record, like molecular or morphological, that would confirm this idea. And so we decided to sample whale bones from the coast of Galicia and Asturias off known medieval whaling ports. Again, there's a problem with these bones because the whales were butchered, so the bones are broken. So as well as the fish bones, they are difficult to identify morphologically. Also, whale collections for comparative analysis are not very easy to find. And we also have modern samples from the northwest Atlantic coast. Nowadays, the northeast population is functionally extinct. The first thing we did was to do a species identification using molecular data. We used a small fragment of the mitochondrial DNA, and out of the 70 specimens that we tested, 68 were actually North Atlantic right whales, so we confirmed that this was the right whale to hunt at the time. And we also got a gray whale and a humpback whale they could have been like caught because they were available, but they were not the main target species of these whalers. And so, given that we had a lot of specimens from these whales, we decided to look into more depth into the genomics of the Northeast Atlantic population, which is now functionally extinct. We screened 40 different individuals, and of those we sequenced 17. Here in this network and in this phylogenetic tree, we only have data from mitogenomes, which is the mitochondrial cells, and we have samples from medieval ports in the Northeast Atlantic, along with modern samples from North Atlantic with no exact record of collection, and modern samples from the Northwest Atlantic from feeding and breeding grounds. And in both of them, we can clearly see that there is a sharp decline in genetic diversity, as all these light blue haplotypes that are present here are only present in ancient samples or medieval samples and not in current samples. We are now looking at nuclear data from these same specimens, and it seems to show us the exact same pattern. So there was a loss of diversity due to whaling. These are just two examples of how you can use ancient DNA to learn from the past, and with this information try to forecast what will happen to species in the face of climate change and have human intervention. I would like to thank my collaborators, my funding, and all of you for listening. Thank you very much.
Thank you, Paula, for this very interesting talk. I'm sure that many of you will have questions. Unfortunately, we're going to have to ask them during the lunchtime, which is actually very appropriate for this talk. So hopefully we'll do that. Thank you, Paula, once again. Now, Helena Amaro, another former colleague here at CIIMAR, PhD in biomedical sciences. She's a microbiologist with 20 years of expertise in biotechnology. She has worked in algal biotechnology for many years, focusing on cultivation, bioactive compound extraction, and valorization of algal metabolites in general. Her research spanned health, food, feed, and environmental applications, and always guided by sustainability. Right now, Elena is Research and Development Manager at Inclita Seaweed Solutions, a company that budded out here from CIIMAR, where she leads the development and commercialization of seaweed-based extracts and ingredients for the cosmetics and nutraceutical sectors. Her work at ISS contributes to advancing sustainable innovations and positioning algae as key resources in the bioeconomy.
Thank you so much, Pedro. First of all, thank you so much. I'm really proud, and Inclita is really proud to be here today. We are really proud to be a part of this CIIMAR. We are born here, as I used to say. We are daughters of CIIMAR, and it's with an enormous pride that we are here today, sharing all the knowledge that we developed at Inclita, that without the knowledge and the advising of CIIMAR would not be possible. So, seaweeds. For many centuries, seaweeds have been a part of society. For many centuries, seaweeds have been seen as a source of food, a source of health. But from the eyes of science, seaweeds are a little bit more. They are a key important, they have a key role in the oceans, in ecosystem services. But for a biotechnological point of view, seaweeds can be more. Seaweeds can be an extremely rich source of bioactives, of health, of skin health, of our pets' health. So Inclita have as a pillar, as a mission, freeing the goodness from the seaweeds, because seaweeds can be an amazing resource that is not fully explored yet. So Inclita Seaweed Solutions have been born here at CIIMAR. We are a biotechnology company, and we really believe that seaweeds are the key to a bio-based future. Seaweeds can be transformed into the next generation bioactives for so many sectors. At Inclita, we make our solutions the perfect match between high efficacy and sustainability, performing and making ingredients that can fit many industries, such as cosmetics, nutraceuticals, food and beverages, pet care, and biostimulants, and even other industries like biomaterials ones. Sharing a little bit of our history, we were researchers here at CIIMAR, and we were born in 2018 officially in a very happy place, and a biodiverse place, in the Laboratory of Biodiversity, where we were inspired by the passion of Isabel Sousa Pinto about seaweeds. And Catarina Isabel could see more, could see a potential of new materials, new ingredients, new improvement, new high value. And together with two great minds that were connected with business and business strategies, they created Inclita Seaweed Solutions. With the time, and Inclita was officially a spin-off, the first spin-off of CIIMAR, and with only possible with the full advising of Professor Vítor Vasconcelos and Isabel Sousa Pinto to fortify our basis, to fortify our expectancies about seaweeds. Later, we were certified for integrity and innovation that put us in a certified way, in a trustability way, that we can produce science to the world. In 2022, we first create our first prototypes here in the CIIMAR. And our team was growing, and we're expanding our potential, we're expanding our knowledge, and trying to put this in the world. In 2023, we had a great strategic growth that allowed us to a market expansion. were funded by Indico and also due with the Plano de Resiliência. We were funded and allow us a better growth, to have more people in the team and to produce even more. 2024, it was a great year for us. We launched our first 3 ingredients for cosmetics, and the team was becoming so big that we need to expand our facilities, and we are at CIIMAR, when we have our office and our own labs. But our story spreads higher this year. This is the golden year. In this year, we are going to scale up and building and starting to produce in our pilot units, when we are going to be able to produce our ingredients and be a mark in the industry. Also, we launched a new ingredient, and at Tinklito, we don't only produce our own ingredients, but we also provide co-development projects, services, and very proud to be with 2 main co-development projects that put us in different countries with 2 very different big industries. So for now, we have our own portfolio. We have 4 bioactive ingredients that are based on seaweeds, in the greatness of the seaweeds. They have different features, but telling this is not enough, because the big thing that differentiates us in the market is our science-based knowledge. So is it not enough that we have bioactive compounds, we prove it and we show it. So every ingredient produced by us ends in clinical trials that we can efficiently demonstrate the bioactivities and the potential of seaweeds. So we have different ingredients in algae wax, that it's a super antioxidant and have regenerative effects to improve the skin renewal. We have an ingredient completely made 100% of chondros, and here we create an exfoliator and with the triple action, because chondros is also a very rich in compounds that promotes the calming effects of the skin and hydration. We have an ingredient that it's completely 100% made in Portugal. It's from seaweeds from Açores. And here we could exploit the benefits of seaweeds from Açores, developing the inalgae azo, that is a powerful moisturizing antioxidant and also has sun protection effects. But we also co-develop. We gather all the knowledge that we have in terms of seaweeds, in terms of biotechnology, in terms of extraction, and we also co-develop services by developing specific ingredients. As we say, your concept, our seaweed solutions. And these allow us to produce tailor-made ingredients for several industries. But in Clita and seaweeds, we want always to be connected about sustainability. And sustainability starts by preserving and respecting the marine resources, the seaweeds. We only use seaweeds that are harvested or produced, respecting all the matters about preserving the oceans. Also, to extract and to obtain our ingredients, we use blue biotechnology methods and a blue biorefinery using green methodologies that allow us to exploit the full power of seaweeds. So we can extract several compounds of seaweeds to be included in the formulation of different products. And of course, that our ingredients can be used to enhance the health and the benefit and the well-being of our society. In terms of global impact, we have spread our message, we have spread the seaweed's capacity, the seaweed's power, the seaweed's benefits all over a little bit of the world. Not only at the national level, by participating in several blue biotechnology clusters when we discussed our main challenges, what we have to do, what we have to explore even more, and even implement in the industry the knowledge of the science, that we believe that it's still a gap that between these two really important connection points, science and in the industry. We have participating have been some interventions with the seaweed agricultures by also making the points that they need to improve so we can improve the bioactivity of the seaweeds. And also, with our growth, we have been recognized, and we are really happy and grateful because of that. We have been in news and interviews at national and international points that allow us to better translate the seaweeds and the science to society. So we have born here, we are here, and the future, I believe that it's not a future without being with CIIMAR. The transition of the science to technology is only possible when we work to the best. So if you want to be the best, we need to be with the better ones, with the best forever. So thank you so much.
Thank you, Elena, once again. Elena is here, and will be here, as she said, also during lunchtime for any questions you might have. So finally, I'm very happy to invite Dora Ferreira to the floor. Dora has been a student here at CIIMAR and eventually went on to do her PhD studies at Nikons in Italy, so a company that's related to natural products chemistry. And she's doing her doctoral studies also under a doctoral network, Marie Curie. She has a bachelor's in medicinal biotechnology from the Polytechnic of Porto, master's in applied microbiology from the Catholic University here in Portugal as well. And during her time here at CIIMAR, on her master's thesis, she worked on natural products chemistry from marine cyanobacteria. Her current work revolves around metabolomics and genomics data for discovering new compounds, and she has switched from cyanobacteria to actinobacteria. So, welcome, Dora.
Thank you. Thank you for the introduction, Pedro, and thank you for the invitation to be here today to talk a little bit about my work in the natural products field, and also the importance of natural products in facing global challenges nowadays. So, I don't think I need to go into any details about the challenges that our world is facing today. I think everyone in this room is very well aware that we have people dying from infections because we have no antibiotics that are active against certain resistant strains. We know that famine is a huge problem in some parts of the world and that we need better and more sustainable solutions, agricultural solutions, and that we need to be more sustainable all around due to the state of our planet. And so there are a lot of strategies that are being worked on to fight all of these issues, but one that has proved time and time again how useful it can be is the use of natural products. So natural products are metabolites that are produced by living organisms like bacteria, plants, fungi, or others, and they are important because they constitute a more sustainable source of chemistry, because these organisms are capable of making structures that we sometimes cannot even imagine possible, and also because these compounds can have high bioactivities that are also selective and that can be of interest to us. So traditionally, to discover these metabolites, a bioactivity-guided approach was followed, which means you extract the metabolites and you test them in a certain bioassay, and if it's active, you separate this extract into smaller fractions, and you continue following the activity and following this process until you obtain a somewhat pure compound that you can know the structure and you can characterize. And in fact, this worked for many decades, and around 75% of antibiotics nowadays in the market derive from natural products. The problem was that the percentage of positive hits in this type of assays was very low, and the same type of microorganisms was usually being used, like streptomyces that are very frequent and easy to grow. And they were being grown in the same standard lab conditions where they might not need to produce the more interesting metabolites. And so all of this led to researchers rediscovering the same compounds over and over again, which led to a loss of interest in this field, especially by big pharma, as they thought that everything that was already interesting had already been discovered. But in the last decades, there have been many scientific and technological advances that have shown that this is not the case, and it is actually estimated that only around 3% of bacterial natural products have been experimentally characterized. So some examples of the advances are that we now have genome sequencing and genome mining tools that identify groups of genes that we call biosynthetic gene clusters that are responsible for the production of certain metabolites. We also have better analytical and computational chemistry tools that allow us to detect, to identify, and to purify better these metabolites. We know that looking into less explored strains that sometimes might have more specific growth requirements can be beneficial. And also, we have a slightly better understanding of regulation. And we know that by mimicking some stress conditions, we might be able to trigger the production of certain metabolites that we don't usually see. And so nowadays, natural products discovery is a very relevant research field again. I first became aware of this field when I was doing my bachelor's internship here at CIIMAR, and at the time I was working on the taxonomical characterization of new deep-sea actinobacteria. And I was incubating them in different temperatures to see which was better for their growth. And I saw that at different temperatures, they were having different colors. And so at lower temperatures, they were having almost this fluorescent yellow color. And so I was told that maybe these temperatures are stressful for the strain, and so it produces certain metabolites in order to help the strain cope with this stress. And I was very fascinated by this concept, and so I decided to keep working on it during my master thesis. So still at CIIMAR, now under the supervision of Mariana Reis, and with the BYT Plus program, I was given 2 strains of cyanobacteria from the Lesh culture collection, and what we were interested in was seeing how the different conditions were affecting the metabolites being produced. So we tested different concentrations of iron and copper because we were targeting metal chelating metabolites, but we also looked at different light cycles and wavelengths because cyanobacteria, as phototrophic organisms, are very influenced by light. I will not go into many details regarding the results, but I can tell you that just by using 2 strains and testing a few different conditions, we saw differences in between all the different conditions, and we saw an overall huge increase of the chemical diversity observed. And one of the most striking things that we saw was that just by having the light on for 24 hours instead of a light-dark cycle, we were able to induce the production of a lot more metabolites, as you can see in pink in this volcano plot. We also identified putative novel halogenated compounds that were specific to only the blue light conditions. Unfortunately, I did not have enough time to pursue this work, but I think that the results really show that this is a viable approach and it can be quite easy to achieve, as you can change any type of condition, and I am pretty sure that you will always see differences in the metabolome. So then I moved on to a PhD position at Nikons, which is a small drug discovery company in Milan, in Italy, where I am mainly supervised by Margherita Sozio. And my PhD project is also a part of a EU-funded Marie Curie doctoral network called Magic Mole Fun, which is fully focused on natural products research and where I have the opportunity to collaborate with people from all over Europe that have expertise in different sides of this field and to also spend time at their labs, which is very beneficial for the science. So now in this project, I kind of scaled up the work that I was doing during my master's, where I now work with 200 strains of Dactylosporangium, which is a rare actinomycetes genus. And for most of these strains, I have complete genomes. I have high-resolution mass spectrometry data from the strains having been cultivated in 2 different media, and I also have bioactivity assay from a very simple antibacterial test. And so what I do is to combine all of these data in order to prioritize, to purify, and to characterize novel compounds, but also to understand more about this genus, which is still very underexplored. So I was able to identify several known compounds in this data set, some that we already knew were produced by Dactylosporangium, while others we had no idea that this genus was capable of producing. And so here we have only a few examples, but as you can see, they have a wide range of bioactivities, from antibacterial, antifungal, antimalarial, anti-cancer, and so on. But still, what we know from this data set is very limited, as we have identified around 16,000 molecules, and we estimate, based on our in-house annotation workflow called Microforall, that around 80% of them are unknown. So out of these unknowns, I prioritized 4 different families of novel compounds that are currently in different stages of purification and characterization, but I can tell you that one of them in particular has high activity against gram-positive strains and particularly against resistant strains. So, as you could see in this data set of around 200 strains, there's still so much left to discover, and this is just a very small portion of the Nikon's culture collection, which is comprised of around 45,000 actinomycetes. And in turn, this is just a very small portion of the biodiversity that our planet has to offer. So I hope that with these examples of my work, I have convinced you of how much untapped potential microorganisms have, that we are now getting better and better at accessing this potential, and that we should definitely be doing it, as natural products are a very viable and more sustainable approach to tackling some of the world's biggest challenges nowadays. Thank you very much for listening, and to everyone that has been involved in all of the work mentioned. Thank you.